Okay, so hello everyone. This is the Structure Meets Function session 39. And so today our talkers are going to be Heikki Saari from the University of Helsinki and Carine de Marcos Lousa from the University of Dundee. Sorry if I butchered that. Um, and these are both active Instruct users, which I'll get onto in a little bit. But first, we're going to cover some information about Instruct-ERIC itself. So some of you may or may not be familiar with what Instruct-ERIC is. This is a platform that countries or specific sort of bodies within those countries pay into so that within the country itself, they have access to Instruct-ERIC funding. This funding enables researchers within for those member areas to actually apply to do readily funded science. So, for example, a researcher can apply to do a project, a short access project of a couple of days or more, without any cost for themselves. And it's as simple as just putting an online proposal where this will go through a standard review process through a number of different technical and scientific reviews. But overall, it's not a very stressful process. It's a couple of pages of information, and a good proportion of these projects actually succeed and go through. So I do really encourage that people look into this, make use of it, and also let their colleagues know about this as well, because we for example, myself, I work at Leeds, and we're aware that even people at Leeds a lot of researchers aren't aware of this platform and how useful it is. So I really do encourage sort of spreading the news. Another note of importance is that if you're collaborating with someone that isn't in one of these member areas, um, and that doesn't necessarily mean that they can't access it themselves. So if you're in collaboration and you are in one of these areas, you can also make use of it for that research. There are a number of other ways that you can use Instruct-ERIC funding. So I've talked to you about the short access visits. There's also a number of opportunities for research and development funding, sort of periodically throughout the year, as well as some internships for some number of months and training as well. So just go onto the site and try and understand what's there. Next slide. Yeah, not much to say here, really. As I said, check out the website. Most of the information is there. If you want to know more, just get in touch with someone from the Instruct team. They're very, very quick to respond and very, very helpful. So with that, I would like to introduce our first speaker. So our first speaker is Heikki Saari. Sorry, yeah, Heikki Saari. Is that the right way to say it?
Yeah, Heikki Saari.
Good enough. So Heikki is an expert in extracellular vesicles. He's dedicated a lot of his research towards understanding sort of the integrity and of these vesicles and how to actually apply them for using them as sort of vessels for treating cancers, transporting some product for actually targeting a cancer. For the purpose of today, Heikki is going to be talking about the research and development funding project that he did through Instruct, which specialized on trying to develop a method by which we can sort of accurately purify and sort of understand the contents of those purification fractions of our extracellular vesicles. So, Heikki, take it away.
Yeah. Thank you. Make sure this is showing properly. So, thank you for having me. I'm Heikki Saari. I work with the Finnish Red Cross Blood Service these days, and the blood service provides blood products to hospitals, companies, researchers from volunteer donors, and also we do research to improve our blood donation process and also develop novel blood products like EVs, which is my specialty. If you're not familiar with EVs, I will briefly go through what they are. They're small membrane nanoparticles that virtually all cells secrete into their environment. In this figure, you can see the main three mechanisms for EV biogenesis. There's the endosomal pathway producing so-called exosomes, and then ectosomes that bud directly from the cell membrane. And then when a cell undergoes apoptosis, they break down into apoptotic bodies. And these vesicles are a very heterogeneous population. Even from single cells, their composition size depends a lot on what kind of cell secretes them, what is their status, are they under some kind of stress. But some common features, they have all this phospholipid bilayer like cell membranes do. And certain entities are enriched, some lipids, for instance, sphingolipids, cholesterol, and certain proteins that are mainly related to the biogenesis process of the EV generation. And then there's certain nucleic acids, mainly RNA, a little bit of DNA is also found, and especially short non-coding RNAs are enriched in EVs, and then there's also other things like metabolites. And you can maybe understand that this makes EVs a very potential target for diagnostic purposes. EVs are essentially a small sample of the cell that secretes them. So there's a lot of research on using EVs for improving cancer diagnostics and other diseases. And then the other aspect is maybe using EVs for therapeutic use, either as such. Some EVs have therapeutic properties, and then some people like myself have tried to load them with drugs because EVs carry these biomolecules from cell to cell. It's part of their job, so they might be very useful in drug delivery. At Blood Service, we naturally study platelets. They are essentially a side stream of blood donation. Mostly blood products that are used are erythrocytes or plasma that have the highest need. Platelets are sometimes given to patients that have clotting disorders, but a lot of it doesn't have a lot of use, so we try to find use for them. Besides blood clotting, platelets do have other interesting properties that are anti-inflammatory, regenerative, that are needed in the wound healing process. So, and part of their function is also to secrete EVs upon activation or also before activation. And these EVs do carry similar molecules as the platelets themselves, so they do have these regenerative properties as well, which makes them a very interesting potential therapeutic product. And platelet EVs might also be especially useful in carrying drugs for cancer treatment, because cancer cells have this natural affinity to platelets. They are dependent on the growth factors and angiogenetic properties of platelets. So some researchers try to make use of this. And since EVs are non-living, they are easier to store than living cells like platelets. And this is a shameless plug, but we do provide commercial platelet EVs through our distributor, Hansa Biomed, if you're interested. So to isolate EVs, and there are several methods available. Most common way people use is differential ultracentrifugation, because EVs are relatively large particles. You can sediment them with an ultracentrifuge, and this is a very simple way producing very concentrated, decently purified samples. It's not very scalable. You would need quite large or many ultracentrifuges to process a large sample volume, and also the sedimentation may cause morphological changes. Another interesting alternative is tangential flow filtration, which is essentially like a filtration. It uses high pore sizes to separate EVs from small molecule impurities. And this is much more scalable, but in TFF, the pores may become clogged, and it's also based on size separation. Another popular method is to use size exclusion chromatography, which is essentially a polishing step to separate EVs from small impurities, and it's very effective at doing this. It is based on size, so if you have large particles such as lipoproteins which are present in blood-derived samples, it cannot separate those from EVs, and also SEC requires a pre-concentrating step. You can't load a very large volume into a SEC column, and it also causes the product to dilute anyway, so you may need to concentrate it afterwards. So from this figure, you can there's a comparison of physical properties of lipoproteins and extracellular vesicles. You can see that there's a lot of overlap between EVs and most lipoprotein species, both in size and density. So based on this, it might not be feasible to separate EVs from lipoproteins, which are present in blood in much higher concentration than EVs are. So because of these reasons, we are interested mostly in using ion exchange chromatography to isolate EVs from blood products. EVs have a very strongly negative surface charge. I think it's because of the phospholipid surface. And because of this, anion exchange can be used as a pretty much universal EV affinity media to purify EVs. Ion exchange is very useful in the sense that at the same time it can remove a lot of impurities and concentrate EVs or whatever you're trying to purify it in a one single step, and it's very easily scalable. Just get the right sized column. They're not very expensive, and this provides basically another dimension to the purification process. Unlike those other methods I mentioned, ion exchange is not based on size exclusion or size of the particle whatsoever. It's just surface charge. So it could be combined very nicely with like a polishing step with size exclusion chromatography to purify EVs in a much more versatile way. And I'll also mention that in our previous work, we found that you can actually also use cation exchange. This works with special tentacle type resin. If you're interested in that, I recommend you check out this article as well. It's from 2023. But on with this, there are some classical methods for characterizing EVs, and the International Society for EVs has published some guidelines to provide some kind of standard for EV researchers, because EV field is still quite young and there's a lot of interest in it. So a lot of researchers are emerging to study EV. So these guidelines help to make other people's research more comparable to others. So we are on the same page on different issues. Some of these methods, the point is to demonstrate that you have indeed EVs in your samples and they are significantly enriched. So you are encouraged to provide some kind of size distribution analysis. We use NTA, which is nanoparticle tracking analysis in our lab. Then you are encouraged to provide some kind of indication of EV marker enrichment. Normally it's Western blotting, but it can be any of these methods. And then, like in the picture on the right, some kind of direct visualization of EVs, so you can show that these are actually intact vesicles. In this cryo-EM picture, you can nicely see the EVs have this lipid bilayer. Some of the vesicles are unilamellar and some of them are multilamellar. This is common. These are vesicles from platelets that we imaged during this project actually. So you can see I think these platelet EVs look exactly the same as all other vesicles I've seen with cryo-EM imaging. So alone, none of these methods provide enough information. You have to provide different aspects to look at your sample to get the whole picture. So a couple words about Raman spectroscopy. This is also something that we have worked earlier. It's a method that is based on vibrational spectroscopy. So it can be used to identify and quantify what kind of functional groups are in your sample, because different atoms bound together vibrate differently based on which atoms are bound to each other and what kind of bond is it. For example, from this study we did earlier, we used Raman spectroscopy to see if we can use it to assess the purity of our EV samples. So, meaning if there are significant differences in the amount of protein impurities with EVs that are purified with different methods. In this picture, we compare ultracentrifugation and ultracentrifugation combined with size exclusion. And the idea is that, for example, this peak, which is 1442, is mostly associated with the lipids. It has this CH2 CH3 scissoring, so groups that are highly enriched in lipids, and then compare that to this peak 1662, which is the amide bond, which is specific to proteins. So combining the intensities of these two peaks, we were able to show that with this method, you can show that, for example, this size exclusion chromatography, you have less proteins per lipid, meaning less protein per EV. But you can see that there's a lot of other peaks as well. You get a lot of information from Raman spectroscopy. So with that in mind, we thought that it might be very useful to use Raman spectroscopy to process the EV purification process. Because Raman spectroscopy is non-destructive. You just measure the scattered light from your sample that you illuminate with a laser. It works with water-based samples, unlike infrared spectroscopy, which is very similar to Raman. Raman spectroscopy provides very detailed quantitative characteristics of your sample, and it can be self-standardized, like I showed in the previous figure. So using it as a method for inline process monitoring, you can use it for these liquid flow methods like liquid chromatography or ion exchange chromatography in our case. We hope that you can maybe directly identify which fractions contain EVs based on the molecular fingerprint that you get from the spectra without doing this laborious downstream analysis like Western blotting and so on. And you can use it to control the quality. Is there some deviations in your sample or EV yield in the fractions that correspond to EVs? So this is the study. It was published last year. This is the abstract from the study. Basically what we did is we got platelets from donors and produced platelet EVs from those, and then we purified them using this monolithic anion exchange column. This was done at the biocomplex purification unit at the University of Helsinki. It's part of the Instruct infrastructure. And we connected this Raman detector to the system that they had. And with that, we were able to measure the Raman spectra along with the UV detector, as you see on the right. With a UV detector, you get a chromatogram much like this, that you have an injection peak, something that comes through that is unbound material, and then when you increase salt concentration, you elute a second peak. But with Raman, we measure the whole Raman spectra every 20 seconds during this process, so we get a lot more detailed information of the whole run during this process. And then we compared that data to this classical EV analysis methods like Western blotting and nanoparticle tracking analysis. And we used 3 different sample types to challenge this system. The first was this standard unmodified sample. Then we doubled the sample load, see if we can overload the column. And then we also tried to add BSA to the standard sample to see what an foreign impurity would look like. And then we analyzed the flow through and eluate to see what happened. So first, this is what the chromatogram looks like. Like I said, there's a flow-through fraction and then elution fractions. Fraction number 3 seems to be the peak fraction. And then we just analyzed with Western blotting how are these EV markers enriched in which fraction, and we find that fraction 3, which is the peak elution fraction, seems to be the most concentrated regarding these EV markers, CD9, CD63, which are the transponin membrane proteins associated with EVs. Regardless of the sample type, this was always the case. However, then we analyzed with NTA how many nanoparticles are found in each fraction, and we find that actually the flow-through fraction contains a lot more nanoparticles than the fraction 3. And with NTA, you cannot separate if it's if the particle is a vesicle or is it a lipoprotein or something else. So this would suggest that some other particles are separate from the EVs. Looking at the physical characteristics of these particles, we find that the flow-through particles are first of all smaller in size compared to EVs. And as a reference, we purified EVs with another method using second ultracentrifugation, so these are very pure. And we find that the fraction 3 particles resemble these reference EVs very closely in their size and also in surface charge. And the flow-through particles had much less negative surface charge than the EVs, which makes sense considering that anion exchange binds negatively charged particles. So the fraction 3 particles are much more negative and they are eluted later from the column compared to these flow-through fractions, which don't really bind the column because they don't have that surface charge. From literature, I know that lipoproteins have a much less negative charge or even positive negative charge. So this all starts to make sense that these flow-through particles might be lipoproteins rather than EVs. So what does the Raman data look like? Here we have color-coded the chromatogram. So the blue is the flow-through fraction and the yellow is the eluted EV fraction. First thing that strikes is that the flow-through fraction contains these 2 peaks in the Raman spectra very strongly, and these correspond to carotenoids. And this in turn suggests that these are containing lipoprotein particles. LDL is what carries carotenoids in the blood. So this also makes sense with our other results. And the other thing you see when you add PSA, which is the spectrum on the right side, you can see that these carotenoid peaks seem lower, and this is because there's a lot of extra protein in there, which makes the carotenoid peaks seem smaller in comparison. So already with this, you can see you can make a lot of deductions on what kind of things your sample might contain. Then using principal component analysis, we wanted to see if we can see differences based on the sample type in these fractions. In this principal component analysis, you can see that these blue circles, which correspond to the BSA spiking sample, you get a clearly separated group in the flow through compared to the others. So there's some kind of difference in composition, and looking at this principal component number 2, which is the separating factor here, we know that this is due to the presence of proteins. Component 1 is mostly carotenoids because those carotenoid peaks are very strong. It's because of some kind of Raman resonance phenomenon that I don't understand, but it makes these peaks very strong and dominate the spectrum. So that's the main component here. When you look at the EV fraction, on the other hand, you don't see any difference in grouping of these spectra. Instead, it seems that whatever the sample was, in the elution fraction, the composition of these samples are the same. So this also goes well with our previous results that the column seems to separate EVs from other components, no matter how much you put into the column or if you add BSA in there. And then finally, we use this PLSDA method to analyze if we can teach this PLSDA model what kind of fractions are coming out of the column. So the way this works is that you teach the model with, for example, the standard run here, which spectra correspond to which fraction. And here the green ones correspond to the EV fraction, the red is buffer, and then the blue is the flow-through fraction. And if you get a value that is above this 0 baseline, the model identifies it either as EVs, or if you look at the flow-through, it identifies it as the flow-through, and then also the buffer. So when you look at the high load run, you can see that it also identifies the EV fraction as EVs as it should, but it also seems to identify some of the flow-through fraction as EVs. So this makes sense if you consider that we overloaded the column. So some of the EVs might come out in the flow-through fraction, and this model was able to see that just based on the Raman spectra and the model we used. And then looking at the BSA run, you see very similarly that the EV fraction spectra seem to correspond to EVs, but then in the flow-through there's something different. It still identifies it as flow-through, but it's not quite as strong than with the other runs, so something is different. So with this kind of analysis, you might look at your spectra run and run data and say, okay, there's something weird about this sample, or in the high load run, you might say, okay, maybe I overloaded the column, I should use a bigger column or less sample. But this is all quite useful. You don't get that much data from just a simple UV chromatogram. This will, I think, in the future be very useful for monitoring the EV purification process, which I think it's a very understudied aspect of this field at the moment. So to conclude, like I said, this Raman spectroscopy seems like a very versatile characterization method. You don't need to do any wet lab work, just analyze the data, which is kind of laborious in its own way, I admit. Yeah, you can identify if there's deviations in your starting material, or if your column isn't performing as it should. And it's especially useful for this carotenoid identification. So if you are using blood-based samples, you can see if you have lipoprotein contaminations or not. And finally, it was kind of surprising how well this monolithic anion exchange column worked. It really efficiently seemed to separate lipoprotein particles from EVs. This is a real problem in the field, because it is difficult, or it is doable, but it's very laborious to separate lipoproteins effectively from EVs. And I think this might be a very useful approach for future studies. And it was very consistent with, as you saw, with any kind of sample. The end product was pretty much the same based on all the analysis we did. And finally, thanks for all the authors that helped do all this, especially Jacopo Zini, who did all the Raman spectroscopy stuff, and funding agencies, and including Instruct-ERIC for funding this and inviting me here. So thank you.
That was really nice. Thank you, Heikki. We've got a couple of, well, we have time for a couple of questions, I think. I noticed that no one's written anything in the Q&A. That's my bad for not telling you it existed. If you want to write a question really quickly, in the meantime, I'm just going to ask Heikki a question myself. So obviously the emphasis on this technique is that you're able to sort of accurately separate out different contaminants or things that you don't want to analyze. Looking at it at a wider scale, how does this purification method, the anion exchange, compare to other purification methods in terms of the yields that you actually preserve? Because you might lose a lot of sample doing this.
Yeah, the yield question is quite complex, I think. I find that it really depends on the sample more than anything else. If you have very pure sample with EVs, you actually may lose a lot of EVs into the column. And if you have a very dirty sample, you get very nice yields. To me, that seems that there's some kind of non-specific binding to the column that the impurities prevent to some extent. So that's something that should be investigated further to understand this mechanism, because yield is very important if you want to use this to produce EVs in a commercial scale or whatever.
We've got a question in chat. Have you tried Captocore resin from Cytiva for purification?
Yes. Also, actually, in this study, we used it to prepare those reference EVs. So Captocore resin is special in the sense that it selectively binds impurities or proteins that are smaller than a chosen threshold. I think it's 700 kilodaltons, or they also have a smaller version. So anything smaller than that is bound by the column, and then EVs come out as the flow through. And this makes it possible to load a lot larger sample volumes into the column. In principle, that is very nice. The negative I find is that it seems to also bind EVs to some extent. If you load a lot of EVs, you can see that the column kind of gets saturated at some point, and after that you get really good yields. So if you have like very dilute samples, I might not recommend this column, but if you don't mind losing a little bit, it's a very nice technique.
Cool. All right, thanks again. So we're going to move on to our second speaker now. So our second speaker is Carine de Marcos Lousa. So Carine is a principal investigator at the University of Dundee. She's also a visiting researcher at the University of Leeds, I believe. And she has been using structural analysis techniques to understand the sort of cargo loading and detachment mechanism of vacuolar sorting receptors. She has done this, some of this research through Instruct-ERIC. I'm not sure if that stuff will be in this talk today. And yeah, go for it, Carine.
Thank you, Louis. Just a little correction. I am not, so this work has been done when I was staff at the University of Leeds and the University of Leeds Beckett, but I am not, it hasn't been done from the University of Dundee, which I moved recently. I'm not a PI at the University of Dundee. So just after this clarification, thank you very much. Hello, everyone. I am going to show what I've been doing thanks to Instruct funding, and I wanted to try and keep this talk simple because I know the audience is varied, but I'm happy to dive deeper into specifics of the structure once if people are interested. So just to explain a little bit about vacuolar sorting. So as you know, plants have a vacuole, which is a multifunctional organelle that plays a role in storage and defense and many different types of cellular processes. And proteins destined to the vacuole are transported through the endomembrane system. And this trafficking is tightly regulated by what we call vacuolar sorting receptors. So vacuolar sorting receptors cycle between early compartments and late compartments to deliver vacuolar proteins into the vacuole in a similar way that is done with mammalian cells and mammalian receptors. So they bind vacuolar cargo in early compartments, traffic to what we call in a plant PVC, which is the equivalent of multivesicular bodies. They release the cargo at this stage, and the receptors are recycled back to early compartments, while the vacuolar proteins are then transferred to the vacuole. So I was a postdoc at the University of Leeds, and I was looking at this cellular trafficking, and I was a little bit frustrated that we didn't have a structure for this vacuolar sorting to try and understand how they function. So I decided to have a look at that more specifically. So just to explain a little bit the structure, as we knew it at that time, vacuolar sorting receptors have a transmembrane domain, a C-terminal domain that has motifs that are important for receptor trafficking, and a cargo binding domain here that is consisting of three main domains. One is what we call PA for protease associated domain, what we call CD for central domain, and three EGF domains here in blue, as you can see here, that are supposed to bind to calcium. What we knew at that time is that glycosylations were predicted to affect the binding, the cargo binding, that there were some disulfide bonds, although they were not identified, and that binding and trafficking is sensitive to pH, but we hadn't a clue about how all these modifications and all these conditions affect the trafficking and binding of the cargo. What I need to specify is that VSR can transport two types of cargo. One of these cargo is called signal sorting, vacuolar sorting determinants here, and they are usually located at the N-terminal domain, and they usually contain a sequence that is NPIR, specific sequence, but also many proteins have a lot of variations in this sequence. So you can find NPIR in some of the cargos, but other cargos will have other types of protein determinants. And one of the model protein for this type of protein is called allorane, and you're going to hear about that in the talk. The second type of cargo is what we call a C-terminal vacuolar sorting determinant, because this sequence that is important is usually at the end of the protein, but there is no consensus sequence. It's mostly hydrophobic in nature. So we didn't understand at that time how one specific cargo could transport many different types of vacuolar cargo and also many different types of determinants here. What we knew when I started is the first crystal structure that has been done by Lu et al. In 2014, where they have expressed the PA domain here in bacteria, and they have identified solved the apostructure, so no cargo, and the complex structure with the peptide here. That is where the sequence is here. But the problem is at this time, they didn't find the binding site of the NPIR specific motif. They found that the arginine 95 was crucial for the binding. And the hypothesis that they did is that they emitted is that the cargo binding domain was across the PA domain and the central domain, which is represented here by a star across the two domains here. So when we started a collaboration with the OPPF, which was the Oxford Protein Production Facility, and they have done these different types of expression for us, and they found that the best expression system was HEC cells, and that expressing the full length VSR didn't work very well, so we only expressed the luminal domain with the PA, the central domain, and the EGF domains tagged with 8 histidine tags at the end, and HBM, so honeybee melitine tag at the beginning for expression in the extracellular medium, where it is purified from. At the same time, in my lab, we have cloned allurane, so the peptide, the signal peptide of allurane, fused to the GFP for visualization. And what we have done is we have also generated constructs of mutation where the NPIR is mutated in APGR to see if our constructs were able to bind to VSR and if it was specific to the NPIR motif. And you can see our purifications here in Combasi are quite clean. And we have used fluorescent anisotropy to look at the binding of VSR expressed in X cells and the recombinant cargo. And you can see here that the recombinant cargo of allurane GFP is able to bind to VSR in solution here when it contains the NPIR, but as soon as you mutate the NPIR into APGR, you don't have any binding. So that means that our system is okay, and we were ready to do some crystal structures. So the first crystal structure we had was the unbound VSR, and this was done in University of Leeds with collaboration with Adrian Goldman, and you can see here the crystals that we got. And that was the first crystal structure we had, where in blue you have the PA domain, in black you have the central domain, and in green you have the EGF domain, and we can identify only, we can solve only one EGF domain. Here you have some amino acids that are relevant for some things that I'm going to say later. So the arginine 95 that has been identified as being important for the binding of cargo, and the histidine here, and another amino acid that are supposed to form salt bridges here. So what we're very happy to see is that we could identify 3 glycosylation sites, and these were exactly the glycosylation sites that were predicted already by other groups. And we also could identify 10 disulfide bonds that have not been predicted, but they were predicted to be present. We just didn't know where they were, so we could identify exactly where they were. What was surprising for us is that VSR was always found as a trimer in the non-loading form of crystal. And you can see here the trimer with 1 monomer here in black, 1 monomer here in light blue, and 1 monomer here in green. And you can see that in this trimer, the PA domain of 1 monomer here in black associates with the central domain of another monomer, and this forms the trimer. And this is very important, this association, for the cargo binding, which I'm going to explain later. But this is this was a surprise that it's always found as a trimer. So then we managed to, we tried to get the structure of the cargo bound form, and we tried a lot with the allurane GFP construct that we had, a recombinant protein, but we were unsuccessful. So we decided to try with a peptide that was previously used by Lu et al. And we managed to get the complex form. So you can see here the superposition of the unbound form here in orange, and the cargo bound form here in green. This is the PA domain, the central domain, and the EGF domains here. And you can see that the structure of the PA domain doesn't change so much, but there are lots of loops here that are changing. And there is an overall rearrangement of the CD domain and the EGF domain compared to the PA domain. So there is a large rearrangement of the structure upon cargo binding. What we also found surprising is that the cargo-loaded form always crystallized as a monomer only. We never found any trimer when the receptor is bound to cargo. The other thing that was very surprising for us, but I expected, is that we found 4 calcium coordination sites in a cargo-loaded form. You can see here calcium 1, calcium 2, calcium 3, and calcium 4. So we were expecting in the EGF domains that we had one calcium binding in each EGF, but we were not expecting to find these calcium coordinations in the CD and across the CD and the PA domain. So the other surprise that we had is that we found 2 peptides bound at 2 separate binding sites. So this is the peptide we used, and you can see that there is the NPIR motif that is the specific motif that is recognized. And so we identified these 2 binding sites. One is named CBS1 here for cargo binding site 1, and one is named CBS2 here for cargo binding site 2. In the CBS1 here, you could see that it is across the PA domain here in blue and the CD domain here in black, while this cargo binding site 2 is only located into the CD domain. The other thing that was nice to see is that the cargo binding site 1 involved a cradle loop here that contained the arginine 95 that I already described before. So it was confirming the results before that these amino acids and other rounds, this cargo binding site were actually involved in binding the cargo, the vacuolar cargo. So at this stage, we hypothesized that one cargo binding site could bind a specific type of cargo, which is SSVSD, containing NPIR or variants, and the other cargo binding site could bind another type of cargo, which is C-terminal vacuolar sorting determinants here. But the question is, why did we have 2 peptides that were bound simultaneously on the structure? And so we looked into trying to understand if the stoichiometry of the receptor binding to cargo. And we used SEC-MALS in collaboration with the University of Dundee. And you can see here when we have only the vacuolar sorting receptor, we have one nice peak and maybe some oligomers that are not very well identified. But you can see that when we add the cargo here, the allorane GFP, then there is a shift towards a size that corresponds to a stoichiometry of 1:1. So one receptor binds to only one cargo. So I will explain later, and I can discuss how we explain that we have actually 2 peptides and only one cargo binds with allorane GFP. But for the moment, I just want to summarize here the results that confirm 20 years of literature, because we have got the 2 structures of the unbound receptor and bound receptor. The PA domain that we found in the structure was exactly the same as the one that was found by Liu et al. When they expressed in bacteria. We also found the glycosylations predicted by Shen. They were exactly at the sites that they predicted. We also found that calcium coordination in the CD and the EGF domains were important for cargo binding. And this was already predicted by Watanabe in 2002. And we also found that the arginine 95 was a residue that was important for binding of the cargo. And this was already predicted by Liu et al. So at this stage, we were very happy. And I just want to explain quickly how Instruct was instrumental in getting all this work done. In 2014, when I started this work, I applied for a Oxford Protein Production Facility Award that was led by Professor Ray Owens, and we got this award so we could collaborate with them to express and purify the first VSRs. We got the first crystals in 2016. Then the diffraction pattern didn't allow us to solve the structure. So we had a few years where we were waiting to find collaborators to help us solve the structure. And in 2018 and 2021, we started collaboration with Isabel Olsen and Rafael Borges in Barcelona because they had developed a new program to help solve the structure of diffraction patterns that were not very resolutive at that time. We managed to finalize the structure of the non-complex form in 2022, but we needed to repeat this. So this is where the Instruct grant was submitted and approved in September 2022. We initially planned to do some cryo-EM and crystallization, but the cryo-EM didn't work at that time, so we concentrated on crystal structure. And this was a collaboration with Andrew Quigley and Rebecca Parr at Diamond. We have confirmed very quickly the structure that we had at the beginning of the non-loaded form, and we got new structures at 3 angstrom. And we submitted a paper, but they wanted us to solve the cargo-loaded form. So the team of Andrew and Rebecca have managed in 6 months to solve the structure of the cargo-loaded form at 2.7 angstroms, which we were very happy with. And then the reviewer asked us to get more data on the oligomers and the validation in vivo. So this is still not published. We submitted in bioRxiv. You can see the link at the end. And we are basically looking at the validation in vivo and answering these questions here. So there are 3 questions that we were asked. Why do we have 2 cargo binding sites in our structures here? And why do they bind simultaneously? So as you have seen by the SECMOS, we can find only 1 allorane GFP bound per receptor. And the reason, Cao et al. Have published that the receptor has 2 binding sites. One that is binding the NPIR, so the SS-VSTs across the PA and the CD domain, and one that is non-NPIR binding site for the C-terminal VSDs involving the CD and the EGF domain. So the fact that there are 2 binding sites on the receptors was known, but in vivo, these binding sites are mutually exclusive. So you can only bind either one or the other type. Our results, because we think we used, because we used peptides, and these peptides are very short, there are only 10 amino acids. We think that this is the serendipity that we could bind at the same time the 2 cargos simultaneously. But this is an in vitro result, and in vivo, this is not happening because these motifs are always bound to a larger protein that is transported to the vacuole. And so we think that only 1 cargo binds to 1 receptor. So our results are also confirming with other results from other groups that the cargo binding site 1 that is across the PA and the CD domain will bind to 1 type of vacuolar cargo, which are the ones that contain the NPIR or variants, like it is represented here from Zhao et al. In 2021. And the cargo binding site 2 that only binds in the CD domain would be binding for the C-terminal vacuolar sorting determinants that are mostly relying on hydrophobic interactions. So at the moment, we are trying to identify a little bit more and explore a little bit more if we are correct or not by mutations specific in these 2 cargo binding sites. The second one is the role of the pH and the calcium. How is the pH and calcium affecting the transport and the cargo binding? So what we have seen is that in the trimer structure, and you can see here a monomer PA domain and a monomer CD domain, where we have superimposed the peptides, you can see that the peptides are actually, they don't have any space to bind in the trimer because the cargo binding sites are masked by the interaction with the other monomer. While when there is the monomer only here, the structure is opening up and exposing the cargo binding sites, which are then allowed to bind each type of cargo. So how is this rearrangement and opening done? this is probably done with the help of the pH and the calcium. And we have come across this paper here by Jos Wick, where they have hypothesized a pH relay where histidine residues are sensing changes in pH. And by sensing changes in pH, they are slightly rearranging the regions for calcium coordination, increasing or decreasing the affinity for calcium. And when they increase the affinity for calcium, this opens up the cargo binding site for the ligand. And we think this is something that is happening as well in the vacuolar sorting receptor, where you have here a crucial histidine 181 that is undergoing a large rearrangement upon cargo binding and is very close to calcium coordination here. So we think the calcium and the pH are actually involved in this pH relay that is rearranging and opening up and exposing the cargo binding sites. And the last one is the oligomerization. Sorry, I'm a little bit late. But we were wondering if the trimer was actually in vivo relevant for the transport. And we came across this paper from 2010, actually, not 14, where they found in vivo higher oligomers of vacuolar sorting receptor. And they found that the TMD and the C-terminal were actually essential for the oligomer formation. In our conditions, we don't have this transmembrane domain and the C-terminal. We only have the N-terminal domain. And by doing lots of different biophysical techniques, we found a mix of oligomers. And we think that in our condition is quite dynamic, and that's why we don't see a specific trimer in our solutions, but we are looking into that. So our working model, because I'm a cell biologist and biochemist, and this is the aim, to understand how the vacuolar sorting traffics, is that in early compartments, calcium coordination happens at high calcium concentration and basic pH. There is the monomerization of the vacuolar sorting receptor. This triggers cargo binding. When the cargo has bound to VSR, it is then trafficked to later compartments here. At this stage, calcium concentrations are low, the pH is acidic. This triggers a calcium release, a trimerization, and a cargo unloading. And the trimerization here allows a signal to be recognized for the receptor to be involving in retrograde trafficking back to the early compartments. And so that is our working model. So lots of thank yous, because I'm not a structural biologist, so I had a lot of help at the University of Leeds, mostly in Jürgen's lab where I was doing my postdoc and I started, and Frank Sobot, who is helping as well with the oligomerization, and Jean, who is helping with in vivo data, Diamonds, Andrew Quigley and Rebecca, who have solved the structure, Adrian, who has helped a lot with the students that were across the University of Leeds, with Sarah Crow, Peter, and my PhD student at the University of Leeds, Beckett, Mahmoud, the OPPF, which was the first funding, Barcelona with Isabel and Rafael, University of Dundee, who has done the segments, and São Paulo, who have done the computation modeling, and the funding here. And thank you very much, and sorry for being a little bit late.
Cheers for that, Carine. It's not your fault. You're not late at all. So yeah, we'll go on to questions now. I can't see any questions in chat at the moment. So whilst we're waiting, I'll just ask a question of my own. You mentioned that there's evidence to suggest in the full sort of length protein that the oligomerization state might be different. So do you think it might always be trimeric and it's just a factor of not having the full length that's causing it to monomerize, or?
Sorry, I didn't, just to clarify, I didn't say that it was different.
Okay.
What I said is that the sorry if I haven't been clear, that the presence of the transmembrane domain and the C-terminal domain might stabilize the trimer in vivo. And the
Okay,
fact
right,
that we don't have the transmembrane domain and the C-terminal domain might explain why we can't find a stable trimer in solution.
Yes, okay, that makes sense, yeah. I guess another question then is, would you benefit from doing time-resolved cryo-EM on this, where you would bind the cargo and capture that conformational change?
Yes, probably that would be great. We have tried cryo-EM before the crystal studies, but at the University of Leeds, but this hasn't been successful, and that's why we forgot about the cryo-EM. But this is maybe I'll submit another Instruct grant and we can continue this work like that. That's a good suggestion. Thank you.
No worries, yeah. Any questions from chat? I guess not. Well, yeah, thank you again, Carine. That was really nice. And thank you, everyone. I think the talks today have been really, really interesting and, well, educational for me as well. So cheers, guys. And I think, John, is that it? Do we just close off at that point?
That's great. Thank you, Louis.
Yeah. All right. And see you guys. Bye.
Thank you, Louis. Thank you, John. And thank you, Sarah.